by Wells Robinson. Please come to the stage. Wells is from the University of Maryland, and he'll talk about modeling clinical and molecular covariates of mutational process activity in cancer. The floor is yours. You've got the presenter. Wonderful.
Hey, good morning. My name's Wells Robinson, and today I'm here to present our approach on modeling mutational processes by using clinical and molecular covariates, and this work has been published in the proceedings. So first, let's talk a little bit about mutational processes. So they're generally grouped into two categories, either exogenous, and this can include UV radiation, smoking, and chemotherapy, or endogenous, and these are processes that happen within the cell, and these generally include defective DNA repair pathways. And it's important to note that a lot of these mutational processes actually cause very specific mutations. And so the first question is, why do we care about mutational processes? So one reason is that a lot of these personalized medicine approaches target defective DNA repair pathways, whether that's PARP inhibitors directly targeting homologous recombination repair deficient tumors, or immune checkpoint blockades being used for tumors with mismatched repair deficiency. Another reason is that mutational processes cause mutations, which are one of the drivers of cancer. So let's look at an example of how we can have three distinct mutational processes that combine to form the mutations in a cancer. So first, we have this endogenous mutational processes that's happening at a low level throughout all of your cells. And then later on, we have an exogenous mutational process, let's say smoking, that occurs at a high rate, but only while the patient is smoking. And then once this tumor has been detected, it can be treated with a chemotherapeutic drug, many of which are cause their own set of mutational processes. And so when we sequence the tumor, we're left with this final cancer genome. And the question is, given this cancer genome, can we reconstruct these three fairly distinctive processes that caused it? And so as a quick aside, for illustrative purposes, I'm going to show examples using these 6 mutation substitutions, but we've found that the 3' flanking base and the 5' flanking base actually matter as well. And so you'll see a lot of using these 96 substitutions. And in fact, using these 96 substitutions, others have identified 30 validated mutation signatures. And a lot of these mutation signatures are actually associated with what we call tumor covariates. And these are just things that we know about the tumor. So this could include, hey, the patient smoked, the age at diagnosis, or known deficiencies in particular DNA repair pathways. And so these mutation signatures have been extracted predominantly by Alexandrov, but also others, using a non-negative matrix factorization approach. In this, you start with a mutation count matrix, where rows are samples and columns are the different mutation categories. And then you want to deconvolve this into 2 smaller matrices, one representing the signatures and the other representing the activity of those signatures in each of these samples. And so one of the issues where these approaches run into problems is with mathematically similar signatures. So up here, I have signature 3 and signature 5. And so these actually have a cosine similarity of 0.83. And so that's really high, because if we were to extract signatures on 2 different data sets and they had a cosine similarity of 0.85, we would just assume that they're the same signature. But we actually know that they're distinct signatures because they're associated with distinct covariates. So signature 3 is actually associated with deficiency in the homologous recombination repair pathway. And signature 5, well, we don't know its exact cause. It's been associated with deficiency in a different DNA repair pathway in at least one cancer type, and it's usually associated with the age of diagnosis. And so these existing approaches struggle to distinguish these mathematically similar signatures because they don't have the ability to incorporate etiology into the model. And so that brings me to the contributions of this slide, of this talk. So in this talk, I'm going to introduce the tumor covariate signature model. And so this is the first model of both mutation signatures and their covariates. So for this model, we're inspired by a less common approach to extracting mutation signatures, and that borrows from the natural language processing field of topic modeling. So in topic modeling, you have a large number of documents, and you want to understand the topics that are present in those documents. And an example of a topic would be a topic for genetics, and this would have a high probability of words like genes, DNA, and RNA. And so there are definitely some differences, particularly in the scale between these two problems. There's a lot more documents out there than there are sequenced tumor exomes, but there's actually quite a few similarities. And so in a given document, we would expect relatively few topics, and similarly in a tumor, we'd expect only a couple of mutation signatures. And there's an established analogy here where documents are equivalent to tumors, words are equivalent to mutation categories, and topics are equivalent to signatures. And so one nice thing about the topic modeling approach is there's a common strategy to solving them. And this is you formulate it as a probabilistic graphical model, and then you perform Bayesian inference. And so the first step in solving a PGM is to define the generative model. And so first I want to take a look at the most common topic model. This is called latent Dirichlet allocation, and describe the generative model in terms of mutation signatures. So the first step is that we assume that we're given these K mutation signatures, and we want to generate the mutations in a given tumor. So the first thing is we randomly choose the exposure, and this is basically which signatures are active in the tumor to which extent. And then for each mutation, we randomly choose a signature from the exposure, and then we randomly choose a category from the signature. And so that works reasonably well, but it kind of ignores one of these key advantages of graphical models. And that's how easy it is to incorporate this observed or latent variables into the graphical model, into the model without complicating inference. So this is in contrast to a method like NMF, where this is possible but would be much more difficult from an inference perspective. And so one example of where topic models have used this approach is by incorporating the author of the document. So in English, there are many words with multiple meanings. One example of this is the word bank. So this can either refer to the financial institution or a riverbank. And so if you know the author is an economist like Alan Greenspan, that helps you figure out that, hey, this word is probably associated with a financial institution. So that brings me to the generative model for TCSM. And so in this case, our goal is to model the association between these observed tumor covariates and changes in signature exposure. So we start off with the same K mutation signatures. But now before we draw the prior, which we used to draw from kind of uniform distribution, now we change the prior based on the observed covariates. And then we just follow the same steps, where we choose the exposures and then generate mutations. And so now let's take a look at what this looks like in practice. So we start out, similar to every other mutation signature model, we have the mutation counts for the tumors. But then we also have a list of covariates and their values for each of the tumors. And so we leverage this information to learn both the signatures, like every other model, as well as a prior exposure weight based off these covariate values. And so now when we want to take these mutations and understand which signatures cause them, we actually use this very informative prior based off of the combination of what are the values for the covariates and what are the prior exposure weights that we've learned. And that provides us with, as I'll show later, more accurate exposures. So that brings me to the next part of this talk, where we're going to identify we're going to show how to use our model to identify significant associations between covariates and signatures and impute missing covariates. So every existing mutation signatures approaches extract signatures, and then they look for enrichments in samples with a particular covariate and a particular signature. And so our model, we're actually able to directly learn this association. And so earlier I showed an easily interpretable prior, and so our model the prior in our model isn't actually interpretable directly. So what we do is we sample from the model. We sample a bunch of exposures, and then we compare between different covariate values for those exposures. And then we provide empirical statistical significance by retraining our model on permuted covariates. And so the next benefit of this model is that once we've learned the model, we can actually take patients where we don't know the covariate, or we just want to hide it for validation purposes, and we can estimate how probable is the set of mutations that we observe given both values. And then we can take the ratio of that, and that gives us the ability to impute these missing covariates. And so that brings me to the last contribution of this slide, and that is a comparison of the performance of TCSM against existing approaches on both simulated and real data sets. So first, we wanted to start with a simulated data set. In this case, we used 4 signatures, 2 of which are similar to each other. And so these 2 signatures are actually the one that I showed earlier. And they can be distinguished because one of them is associated with a particular binary covariate, similar to the generative. We actually used the generative model that I described earlier. And so we identified 2 main issues with the existing approaches. And the first one is in the problem of model selection. So the existing approaches really struggle to distinguish between these similar signatures. And so more often than not, they identified k equals 3 as the correct number of signatures, while our approach identified k equals 4 on most of the data sets. And our approach that didn't use the covariates actually performed even worse than the NMF-based approach, showing that this improvement is due to the incorporation of the covariates. And next, we wanted to see what would happen when we forced the approaches to use 4 signatures. And what we found is that our model did significantly better in terms of distinguishing, hey, this mutation came from signature 3 or this mutation came from signature 5. And so you can see this, we used mean square error across a large number of samples, and we used 50 data sets. So next, we wanted to test this model in a real data set. And so given our interest in signature 3 and homologous recombination, we actually looked at 900 different breast cancer samples from TCGA. And so the first thing we did is we wanted to experiment with using different covariates to represent homologous recombination efficiency. In this case, we focused on using the LST count, which is the number of chromosomal breakages greater than 10 megabases. This is a continuous value. And then we also looked at the inactivation of genes in the HR pathway. And we compared them using held out log likelihood. And so what we actually found is that the LST count performed the best. But a subset of the genes in the HR pathway, BRCA1, BRCA2, and RAD51C, performed almost as well. And given how much easier it is for our downstream analysis to use binary covariates, we decided to focus on this. And so these are the covariates that I'll be using for the rest of the talk. And so using these covariates, we identified 5 known signatures, and we found only one significant association, and that was between the HR covariate and the signature most similar to cosmic 3, which is the one it's already been associated with. And so next, we wanted to test our ability to impute the status of the HR covariate on real data. And so we used the imputation framework that I showed earlier, where we trained the model on a subset of the data, and then for the test data, we try to impute the covariates using the model and the mutation counts. And so you can see up here, we do a very good job of first separating the known BRCA1, BRCA2, RAD51C biallelic inactivation events from just the other HR genes that aren't known to cause signature 3. And then we also did a good job of separating from the wild type that didn't have any inactivation. And then we actually have some interesting evidence that these are likely to be HR as well, based off of the LST count. And so the next thing that we wanted to do is we wanted to benchmark on real data. And so in this case, this is actually a little bit difficult because we don't have any ground truth. And so what we decided on is that the model that provides better exposures, more predictive exposures of something, is the better model. And so in this case, we decided to train a classifier on exposures from the models, and we wanted to try to predict homologous recombination. The same covariate that we're using to train the model. So obviously in the test data, we actually followed the imputation approach. And so we were able to evaluate using the area under the precision recall curve. And we can show that TCSM does better than both TCSM without the covariates and the somatic signatures, which is the NMF-based approach. And so the next thing is, how can we apply this model to more data sets, or to more covariates? And so one thing in the field is that generally, these models have been trained on cancer type specific data. And so that's in large part because there's very different signatures across these cancer types, and that can confound the analysis. And so we think using TCSM, we can use cancer type as a covariate and infer the signatures across these cancer types simultaneously. And then just as an added benefit of this model, we can also then impute the cancer type using observed covariates, which is an active area of research in the field. And so as a test of this, we decided to focus on melanomas and lung squamous. And so we decided on these two cases because of previous reports that some lung squamouses in TCGA are actually misdiagnosed metastatic melanomas. And because there is a particular dinucleotide polymorphism, CC to TT, that isn't included in the 96 categories, but is known to be associated with melanomas. And so as you can see, we found 3 lung squamous cells that were really as strongly as most of the melanomas to be melanomas. And so it just so happens that these are the 3 with more than 15 of these dinucleotide polymorphisms. So I would like, in conclusion, hopefully today I've shown you that integrating tumor covariates is important, particularly for distinguishing between similar signatures. And some of the future directions we're interested in are these pan-cancer signature extraction, and modeling covariates affecting the signatures, and also leveraging some of these topic modeling approaches. So with that, and I would like to acknowledge my 2 co-authors, the entire licensing research group, my other advisor, Eitan Rupin, and the Cancer Data Science Lab. And if any of this is interesting, I would invite you to submit a paper to the Pacific Symposium on Biocomputing, which is in Hawaii. So that alone should hopefully appeal. Thank you.
Thank you.
I was at 17 minutes.
Okay. So we have time for one quick question.
Yeah. So Steve Rosen, Duke NUS Medical School, Singapore. That's really nice work. Could you comment on the runtime for this and what kind of algorithms you're using for computing these factorizations?
Sure. So we actually leveraged, there's a R package called structural topic models, and they used a variant of variational inference to solve this.
In the runtime?
In
computational resources needed?
Minimal.
Minimal.
Yeah.
Do I have time for another question?
Yeah.
Okay.
So quick,
have you
do another one? Yeah.
Is there somebody else? Yeah.
Yeah, you're fine.
Okay. So have you compared your stuff looking at homologous recombination deficiency to bespoke methods like HR detect?
Well, so HR detect leverages both single nucleotide polymorphisms and quite a few whole genome sequenced. And so we didn't do that comparison.
Okay, great. Really nice work. Thank you.
Thanks.
Thank you again.